Hey guys, Miss Cote here. This is the last in a three-part series on the immune system. In part one, we looked at innate immunity, the nonspecific immunity that we have from birth. In part two, we looked at how B cells and T cells in your adaptive immune system develop. Now let's keep looking at adaptive immunity, focusing on the specific way these cells respond when they are activated by a pathogen. So let's get started. B cells and T cells, because they respond to a pathogen differently, are divided into the humoral immune response and the cell-mediated immune response. The humoral immune response involves the B cells. The humors is an old term that was once used to refer to the fluids in your body, primarily the blood and the lymph. The B cells, which patrol the body using those vessels, respond to an antigen by producing antibodies to neutralize pathogens in those areas. The T cells are involved in the cell-mediated immune response. This happens in response to your own body cells becoming infected, for example, with a virus. The T cells will find those infected cells and destroy them, thereby not allowing the virus to be released to infect other cells. There are actually two types of T cells, helper T cells and cytotoxic T cells, each with different functions. First, let's talk about a helper T cell. While the helper T cell does not actively fight pathogens, when it's activated, it will activate a response in both the humoral and cell-mediated pathways. This cell is extremely important in maintaining your adaptive immune system. Helper T cells are activated by what's called an antigen-presenting cell, which is a cell that has encountered a pathogen and presents a piece of that pathogen, the antigen, to activate a response. Examples of antigen-presenting cells can be dendritic cells, macrophages, B cells, or even your body cells. They have a special protein on their surface to display antigens. These are referred to as MHC, which stands for Major Histocompatibility Complex. There are two classes of MHC, Class 1 and Class 2, and the response will depend on what class MHC the antigen is being presented on. So here on top, we have an antigen-presenting cell that is engulfing a pathogen. As it breaks it down, it displays pieces of it, antigens, on its MHC. In this case, it is MHC class 2. This particular helper T cell recognizes an epitope with its receptor. To strengthen the binding, the helper T cell also has an accessory protein that will bind to the MHC. This accessory protein is referred to as CD4, and CD4 always pairs with MHC2. These two cells interact, communicating with each other by secreting signaling molecules called cytokines. This activates the helper T cell and causes proliferation. The proliferation of the helper T cell produces clones, which all have the exact same receptors for the antigen. These cells can now either activate B cells to secrete antibodies or to activate cytotoxic T cells to destroy infected cells, or both, depending on what the body needs. In the cell-mediated pathway, cytotoxic T cells, which are also called killer T cells, are the ones that directly attack and destroy your own infected body cells. To become active, a cytotoxic T cell needs to be activated by a helper T cell. So let's look at how this happens. Here we have an infected body cell that's displaying an antigen on its MHC. Body cells have MHC class 1. The cytotoxic T cell, with a receptor that matches that particular antigen, will bind along with its accessory protein called CD8. Just to make a side note here, the way I've always remembered what goes with what is that the product of the MHC and CD proteins must be 8. So MHC1 will always match with CD8 and MHC2 will always match with CD4. If that helps, great. If not, you can find your own way to remember. All right, back to the diagram. After binding, the cytotoxic T cell will release molecules called perforin, which will go to the cell membrane of the infected cell and create pores, or openings in the cell. Then a second type of molecule called granzymes will be secreted, and these molecules will enter the infected cell, and between fluid and ions rushing into the cell and the granzymes digesting the cell from the inside out, this will cause the cell to undergo apoptosis and be destroyed. This is an infected cell's way of protecting the neighboring cells around it from getting infected themselves. Kind of altruistic. Now let's take a look at the B cells and the humoral response. B cells also require activation by helper T cells, but unlike the cytotoxic T cells, the B cells also take in pieces of the antigen that it binds to and can act as antigen-presenting cells all by itself. This should look familiar to you since you just saw it a few slides ago. We have an antigen-presenting cell that's engulfing a pathogen, displaying an antigen through MHC2, binding to a helper T cell along with the CD4 accessory protein. What's also happening is that this B cell has come across the same antigen and binds to it with its matching receptor. The B cell has the ability to take in some additional antigen molecules through receptor-mediated endocytosis. That was back in chapter 7, by the way. The B cell also acts as an antigen-presenting cell and displays the antigen through its MHC2 protein. This matches up with the activated helper T cell, which secretes cytokines and activates the B cell. From there, the B cell will proliferate and make clones of itself. While some clones become memory cells, the plasma cells will begin to secrete antibodies, which are specific for the same antigen that initiated the response in the first place. So if antibodies don't go out and actively kill cells, what's their function? Antibodies tag or mark pathogens for either inactivation or destruction. They can do this in a few different ways. In neutralization, the antibodies bind to antigens to block it from binding to a host cell. This would be good for a virus, since a virus needs to enter a host cell to reproduce and make copies of itself. Neutralization would prevent that from happening. Opsonization is where antibodies would bind to antigens and tag it for eventual phagocytosis by a macrophage. This is good for pathogens like bacteria that don't necessarily infect our cells, but could potentially secrete toxins to make us sick. A third way for antibodies to work with the complement system, we briefly mentioned the complement system in the innate immunity video. Here we have antibodies binding to antigens on a pathogen. This brings in the complement system, which will cause destruction of the pathogen by poking holes in its membrane, causing water and ions to rush in, swell up, and burst. In summary, we've seen how the specific lymphocytes of the adaptive immune system respond to pathogens. They even use players from the innate immune system in the process as well. So we do see crossover between the two parts. Note that all three of these lymphocytes, B cells, helper T cells, and cytotoxic T cells, have the ability to make memory cells in response to a threat. And this means that all three types of cells can produce both a primary and a secondary immune response. Remember, we learned about that in the second video. Remember the graph with the blue and red lines. So that basically covers the immune system. It's an extremely complicated system involving many players. So I hope I simplified it a bit for you. Hopefully, we'll have some time in class to explore how the immune system functions in certain situations, like the rejection of tissue transplants and allergic responses, and others. And until next time, be smart and be safe.